Well, good evening and welcome to the Logos Research Associates live stream broadcast. I'm Jay Siegert. I'm going to be your host this evening. I'm also the founder and director of the Starting Point Project. Again, host for the evening. There's no need to panic. Good thing. But if you're not familiar with our group, Logos Research Associates, let me read you a little paragraph of our background. It's a fellowship of scholars and scientists that are committed to the upholding a high view of science and a high view of Scripture. We sincerely believe that scholarship, logic, and a scientific method can and should be used to show that the Bible's historical claims are both credible and ultimately superior to the secular model of origins. Logo sponsors many creation research initiatives and projects involving many different experts and scientific disciplines, all working together for the goal of increasing the explanatory power of the biblical creation model of origins and of Earth history. The goal of our live stream broadcast is to provide you with the opportunity to hear from top researchers who will be conveying cutting edge research for the Genesis creation account. Before I introduce our speaker for this evening, just join me in a short word of prayer. Dear Heavenly Father, we just thank you so much for these times you've allotted for us to meet together. Thank you for the technology that we can use. Thank you for all the speakers that we have and the gifting that you've given them to convey what you've put on their hearts, and just the exciting discoveries that they keep making, and just further and further confirmation of the biblical worldview. We just pray that you would be blessed and honored in all that we do, and pray these things in Jesus' name. Amen. Just before I introduce our speaker, just really quick background. There are a lot of creation speakers out there and researchers, and nobody knows everything. A lot of them specialize in something. I happen to specialize in everything, which is kind of true, which means I need to know a little bit about everything, which means I generally don't know tons and tons and tons about any one area. So we rely on experts to be doing the actual research, and then we glean from their research and put it into our presentations. And in a sense, we need to trust the work they're doing. And it works out, I think, very well because they are very intelligent. They're doing great research, and they also have a moral obligation to be honest with their findings, which means if they discover something and we need to go a different direction, we do that. So I think it's pretty exciting where we can keep refining our information and make sure that the rest of us who aren't doing hands-on research, we know that we're getting the latest and conveying top-notch information. So we're going to be talking about some fascinating things tonight, and I think it's going to involve some shifts, some small, some big shifts in some things that we had thought before, but new information comes along, and it's very exciting. So with that, let me introduce Dr. Rob Carter. He's a Logos Research Associate. He's also a PhD marine biologist. He's a senior scientist and speaker for Creation Ministries International. Great organization. I actually did some speaking for them many years ago for about a year. I left, and then they really took off after that. I don't know if there's a correlation there or not, but they're based out of Atlanta, Georgia. And Rob is currently researching human genetics and other issues related to biblical creation. He's going to share some cutting-edge research that exposes the flaws of the evolutionary interpretation of chimp and human genetics. You can visit his personal website at biblicalgenetics.com. And then also for Creation Ministries International, they have a very, very complex website, creation.com. That's a great one. So, Rob, welcome to the broadcast.
Thank you, Jay. That was some very nice things to say, and we do miss you over here. Okay, let's just jump right in then. Can I do that?
Yep.
All right. Well, everyone, I want to say thank you for joining us, and for those people watching later, thank you for watching. Thank you for Logos for the opportunity. This is something that I've been both looking forward to sharing and dreading at the same time. I am going to get my button correct there. There we go. I'm going to be discussing my latest research on human and chimpanzee genetic similarity and dissimilarity. One of the reasons why I'm dreading this is, and not so much for the embarrassment, I am going to contradict some of the things that I've said before and some of the things that I've written before, but I'm a big boy. I can handle that. I understand that anytime you publish or say anything as a scientist, you're opening yourself up for contradiction. That's how science works. We advance by showing people in the past were wrong. And so that's just a fact of everything, and I'm not embarrassed about it. It's just a little humbling sometimes. What I'm afraid of, though, is that I'm going to have to contradict some of my fellows, and I don't enjoy that at all. But again, as scientists, I've spoken to all the people involved. They know where I'm coming from. There's nothing to be surprising here. But the ball advances down the field sometimes, and sometimes we have to let go of older ideas. Something else I'm concerned about is that I know that some evolutionists are tracking what we're saying on these subjects, and they might misunderstand some of the things that I'm saying, or saying, oh, look at that, they agree with us, evolution is true. No, that's not true at all. But I am going to bring the similarity arguments a little closer to what the evolutionists have been saying for a long time, and a little further away from what the creationists have been saying, myself included, for a long time. In way of background, I am a marine biologist by training. I'm actually a coral reef ecologist. That doesn't sound like a human chimpanzee genetics expert, but while I was studying corals, I ended up studying a group of proteins in corals, the green fluorescent protein gene family, which includes green and red and other colors of fluorescent proteins. But when I started, I didn't know what the colors were. I thought they were a chemical. When I figured out that they were a protein, then I realized there's a gene behind it, and I ended up in the genetics lab. And when I stole the genes out of corals and engineered them into fish, that's when I was awarded my doctorate, basically in genetic engineering. And since then, I never looked back. I mean, as soon as I was done with my doctorate in 2003, I guess it was, I went straight to the Human Genome page and hit download. And I've been writing computer programs and analyzing DNA ever since. And I do a lot of other things also, but genetics is a passion of mine. I just want to know more about DNA and genes and how things are passed on. But we're going to get into now a very controversial subject. The question is human and chimpanzee genetic similarity. But I want to share something with you before we even get into it. Something that I learned myself in graduate school. Now, I've heard these called type 1, 2, and 3, and I've also heard them called type A, B, and C. But I like type 1, 2, and 3. There are 3 different types of experiments. The gold standard, the thing that all scientists strive for and very rarely achieve, is a type 1 experiment. That's an experiment that can split the difference between 2 competing hypotheses. It's not easy to do that. Usually you can address one hypothesis and not another hypothesis, or a hypothesis neutral on that. And that leads to a type 2 experiment. This is something that supports one hypothesis or contradicts one hypothesis, but has nothing to say about another hypothesis. And a type 3 experiment is actually sad because all scientists do this. This is something actually that gets published sometimes. It's an experimental result that has nothing to do with the hypothesis. It's just a bad experiment. It was set up wrong, or assumptions were made. And type 3 experiments are everywhere. Type 2 experiments are the most common. Type 1 are very rare. I say that because the human chimpanzee argument is a type 2 experiment. It doesn't matter to the creationist. We have a God who created. He could have created us with as few or as many differences as he chose. So whatever the answer is, that's the answer. Okay, I can take that. The evolutionists, though, they're in a little bit of a box in that they can accept a lot of differences, but that would require them to back up the timeline, and the paleontologists won't allow the great apes in the time of dinosaurs. So they can't back it up too much. You can't say like humans and chimpanzees split 17 million years ago, because that messes up all the paleontology. But there's a huge range that they can accept, huge range that we can accept. It honestly really doesn't really matter that much. But we get to ask the question then, how different are we really? Are humans and chimpanzees 99%, 98%, 97, 96, 95%? I've heard all those numbers coming from the evolutionary community. The typical answer you have from the creationist side is 85%. How do I say this? Honestly, I've never been very comfortable with that number. I don't know if that's biologically realistic. 85%, that's a lot of difference. And if you look at chimpanzees and all the other species in the world, chimpanzees are most like us. They kind of look like us, and on some levels they kind of act like us, and they have the same food requirements and the same temperature requirements. They are basically spiritually alive as a box of rocks. They are not intelligent. We're qualitatively very different from them, but quantitatively, I would expect on first principles that humans and chimpanzees should be similar on the inside because we're so similar on the outside. So I think the creationist community says, yeah, that we should be similar. The evolution community says, yes, we should be similar. But no one actually knows. No one, before the numbers were run, no one could make a prediction. Back in the 1940s, when we started sequencing proteins, everyone expected that human proteins and chimpanzee proteins would be different because they're different species. When they started finding proteins that were the same, that was actually a challenge to the evolutionary community even. Why are they the same? That doesn't make any sense. They should be different. Then DNA comes in, and then in the 1970s, we started doing DNA hybridization experiments where you mix human and chimpanzee DNA or other species in a test tube and warm it up and cool it off. And as you shine light through the test tube or the cuvette, the solution becomes clear at a certain temperature as it warms up, and it becomes more opaque as you cool it off. And by doing these association studies, you can estimate how similar two sets of DNA are. In fact, one of the people who pioneered this between humans and chimpanzees, I have his book on the shelf behind me. This is where is it? Right there. Oh, I shouldn't have done this because now I can't find it. It's right behind me on the shelf. It's the phylogeny and classification of birds by Sibley and Alquist. Now, Alquist at the time was an evolutionist, but once he realized that his experiments, of course, they show high similarity. 98% was the conclusion, because we have a lot of similar DNA. But what his experiments didn't sequence or didn't estimate was the areas that wouldn't align. And so what he concluded was that my evolutionary understanding is wrong, and this is one of the things that led John Alquist to be a young Earth creationist. We interviewed him in Creation Magazine. I've spoken with him. He's now dead, but he had a big conversion experience in his life, partly because of the scientific data. So this is a big deal to a lot of people. Before I move on to the real meat of the argument, I want to also talk about a frustration I have with the field of baraminology. Now, baraminology is a creationist concept. Baramin or Baramin is a made-up word that is joining together of two Hebrew words, bara, to create, and min, type or kind. Because in Genesis, God said he created things according to their kind. He didn't say how many kinds. He didn't say what the diversity within kinds was or how far apart kinds were, but we have a general idea that God created groups of organisms reproduced within themselves and not with other things. But in the field, there's no metric. There's no statistic you can say like, oh, the Cartesian Gaussian coefficient squared equals 0.32221. Therefore, these two things are two different created kinds. We don't have that number. And so we see that some baramins are more similar than to others. I mean, consider all mammals are more similar to each other than any mammal is to a fish. Why? Because God created hierarchically. And in that hierarchical scheme, some things are closer together than others. So humans and chimpanzees, they could be really close or really far apart. I wrote an article with my friend Matthew Serhati. We published in the Journal of Creation. It's called Hierarchical Clustering Complicates Baraminological Analyses. All we're saying is that if God created hierarchically, then some distinctly separate creations could be very similar. Other distinctly separate creations could be far apart. So there's no single measurement that anyone can use. You can't just say it's equal to whatever that number I quoted earlier that tells us they're two different created kinds or the same created kind. Now, I love baraminology. I love studying the discontinuities between groups. It's really fun, really interesting. It's just a little frustrating, and that directly applies to the human chimpanzee question. Now, about a year and a half ago, I really stuck my foot in it. I heard about these series of videos that some group of evolutionists were making against one of my friends, Jeff Tompkins, over at the Institute for Creation Research, who's a geneticist. There's not a lot of creationist geneticists out there, and so I was worried about it. And I watched the video and said, wait a minute. Basically, my first video that I produced, I said, okay, here's what they say, and here's what he says. Here's my opinion. And I got a lot of flack from that because I didn't address what was in the video that I claimed to be addressing. But that wasn't my purpose, but I got a lot of flack. I ended up making 5 videos. Some of these are very long, some not so long, on this issue. Back and forth between myself and several evolutionists, trying to get at the meat of the argument. One of the things that happened was that when Jeff Tompkins started doing human chimpanzee comparisons, he did not want to use the original chimpanzee genome, and there's a good reason for that. The chimpanzee genome had been humanized. What they did was, instead of spending $3 billion making a chimpanzee genome like they spent on the first human genome, they lightly sequenced the chimpanzee genome and hung the pieces on the human genome to assemble them. Well, wait a minute, that's assuming common ancestry. That's assuming the pieces are where they should be, and it caused all sorts of problems. Well, actually, the part of the human genome I learned later was faked. I didn't know that. They actually filled up some stretches with assumed sequences. The average of that is this, so they just put a whole bunch of that in there. So when they then did the chimpanzee, they also lined it up possibly with fake sequences. And so that meant that he wanted to do something different. And what he ended up doing was, in one of his papers, he started with basically the raw sequences that come out of the machine, not the lined up sequences that they use to build a genome, but the raw data. And he said, okay, I'm going to use this because it has been humanized. And he ran his experiments, and that's where we get one of the claims of about 85-ish%. And that's what these evolutionists were criticizing. But as I'm going through this, I'm very confused. And I'm listening to the arguments, and well, I made some mistakes because I started addressing an argument which came from a different paper that I wasn't talking about, and they got confused about what I said, and on and on and on. And then I realized an error that the evolutionists were making and an error in the original paper. And one of these articles, one of these videos I produced, I said, hey, everybody stop. Everyone's making errors. And I brought up this, James 3:1. This is a verse that should keep all scholars up at night. Not many of you should become teachers, my brothers, for know that you who teach will be judged with greater strictness. Or we who teach. That's scary. That is really scary. Someone who opens up their mouth and claims to know what they're talking about, you get a double judgment. Ouch. That is terrifying in one sense. But then again, we're all human and we all err. And scientists are human. We're bigoted, we're biased, and a lot of times we're just stupid. And we're trying to do these things that we don't understand, and we're on the edge of our abilities and the edge of our understanding, and well, mistakes are made. I published a paper in our Journal of Creation last November, I think it was, a year ago, November, reassessing human chimpanzee genetic similarity. And I discussed some of these issues then. I should probably have another paper with what I'm about to show you, but this is a year or so ago. And one of the things I realized after really digging in deep was that when Jeff had run his first experiment, he had tens of thousands of data points. And the assumption was, man, if I have tens of thousands of data points, I have a good average. And that's generally true if your data are what's called normally distributed. If they follow sort of like a bell curve shape, then you can take the average and everything's cool. But when I plotted the data, and no one else had thought to do this, when I plotted the data, I saw that there was a lot of chimpanzee sequence that was like 98% similar to the human. And a lot of chimpanzee sequence that was like, 68% similar to the human. He took the average of that and got about 85%. But there's no data in that range. It's high and low. There's nothing in the middle. In statistics, we learned that you cannot take the average of something like that. That's an illegitimate average. It's sort of like, think of this analogy. Imagine that there's a field of flowers. And there's 50-50, the flowers are purple and white. And there's one species, purple or white, and that's the only color of flower. And you wanted to say, hey, what's the average color? Well, there is no average color. It's purple or white. But if you took a machine and harvested all those flowers and pulled the pigment out, now there's purple flowers have pigment, the white flowers have none. And then if you calculated the average pigment content of the flower, you would say, oh, the average flower must be blue. But there are no blue flowers. That's essentially what happened. Now, it's an honest mistake. I mean, I have done this. A lot of other people do this. It's just when you take a lot of samples, you have to test to make sure your samples are normally distributed. A lot of people don't, and that was done a long time ago, and that's one of the things that nobody noticed. So, knowing that, I'm looking at this data, and I'm saying, what is the similarity? I have two bunches here, and then I realize, I don't know that machine randomly pulled data from the chimpanzee genome. What if the machine like fixated on highly repetitive DNA, which is the stuff on the left, the low similarity stuff? What if it just fixated on that and really pulled that up? Or what if there was a part where they filtered the DNA in a chemical solution or a gel bead or something like that, and that distorted that kind of DNA, maybe pulled some DNA out that should be in the middle? Or what if after the fact a human curated the data or use an algorithm to separate out the data, and what we're getting is not actually the stuff that just came out of the machine, something that's been curated? Well, I don't know the answer to that. Or it might be that there is some DNA that's highly similar between humans and chimpanzees, like 98, 98%, 99%, and some that's not. Well, that's actually going to be true, but hold on, we'll get back to that in a second. More fuel was added to this fire by a guy named Richard Buggs over in England. He published a blog post where he said that the human-chimpanzee similarity, when you factor in all the data, is probably in the mid-80s. Oh, just like Tompkins had said. But then, to make it even worse, he published a paper with Josiah Seaman. This is Josiah's doctoral work about a new computer program Josiah wrote called Fluent DNA. Now, this paper is beautiful and amazing and really fun to read. And what Josiah did was he developed all these new ways of visualizing DNA similarities. So looking at stripes and color changes and things as you do all these different ways of looking at the differences, and it's absolutely fascinating. But their paper, if you look on I don't know if you can read it on the screen, but on the bottom there, I'm reading 97.96%, 96.66%. They're getting really high similarities. And so all of a sudden, everyone says, oh, look at that. Even Richard Buggs doesn't believe it's 85%. He's now saying it's like 97%. No, because if you parse all those numbers, there's a lot of letters that are left out. That's the high similarity is only the stuff that aligns, not the stuff that doesn't align. And so that question is still there. What aligns and what doesn't? And that's a massive problem. So what I'm suggest to you, and I think going forward, if both sides acknowledge what I'm about to say, I think it's going to help everybody. We actually need three numbers. We need the percent similarity of the alignable regions. That is what John Alquist did in the 1970s. This is what the estimates were in the 1980s and 1990s. This is what people talk about now. Similarity of only the alignable regions. And you know what? That's going to be like 98%. It just is. But then I want to know the total percent similarity. When you roll in all the stuff that doesn't align, that percent similarity is going to drop. Probably 85%-ish. Maybe 90%-ish. It's somewhere in that region. It's nowhere close to 98%. Maybe it's 95%. I'd be happy with that also. But it's lower than the 98% we hear so often. But honestly, those two numbers are irrelevant. Really, for addressing this argument, those don't matter. It's the third number that's really important. And that is, how many mutational steps are necessary to drive the differences between humans and chimpanzees in the evolutionary 6.5 million years since our common ancestor? That's the number we need to know. And that number is actually hard to come by. One reason is that it's expected something like 99.999 something percent of all new mutations will be lost. Second, a lot of mutations are deleterious to the organism, so natural selection is going to be removing them. It, but maybe with some mutation rate and 6.5 million years, that's a long time, you can get the number of differences that we see. But again, it's a type B experiment. Even if the evolutionists can, a type 2 experiment, even if the evolutionists can say, we can explain these differences, the creationist says, well, we don't care. Great, evolutionists, we're happy for you, but that does not contradict our position here, because we don't make a prediction on this one thing. There's other things we do predict. This one we don't. What's the difference between humans and chimpanzees? It doesn't really matter. But even that's not super important. What's even more important is the integration time. And that is, if a new mutation arises, a new gene family, an inversion, a duplication, a deletion, a translocation of DNA from one place to another, that's going to influence how genes interact with one another. A long time ago, I learned that the difference between human and chimpanzee genomes is not the sequence. It's the gene interaction networks. It's which genes are turned on at what time, which genes are turned on how much or how little, and how the genes interact with one another. Those things are vastly different between humans and chimpanzees. That's, well, something I can't even count. There's something above my head because I really don't even understand it. It's really deep, crazy kind of stuff. But if you're going to take a human, if you're going to take a human chimpanzee common ancestor and split into humans and chimpanzees, you have to have tens of thousands of genomic rearrangements. You have to move genes around. You have to change the coordination of entire genetic pathways. That is extremely difficult, and we would expect that almost all those changes are going to be detrimental to the organism. A paper came out in Science just last week, Inherited Genome Instability. They're talking about one particular class of segmental variations, which is a copy number variation if you like, that causes childhood cancers. Yeah, you can't just change DNA. You can't move everything around at random. There's a lot of bad stuff that can happen. The genome is not infinitely mutable. So if you want to cause that many mutations, which is the number of differences between us times many thousands, because most of them will be lost over time, you're talking about a very large mutation burden. You're talking about a mutation burden that should be extinction level. But that's another issue for another day. I'm just putting a warning flag out there for the evolutionists that they've got some more things to account for, and they pretty much have assumed for the last 150 years that life is really simple. And it's just not. So, now for some data. Or before we get to data, a background of one particular claim. Now, I chose this as my test case because I can kill more than one bird with one stone. Let's look at what's called the chromosome 2 fusion hypothesis. The evolutionary story is that two small ape chromosomes fused in the early human lineage. Now they're putting it just over 1 million years ago. And those two chromosomes fused to make chromosome 2 in humans. I'm going to tell you something else. Just like the 85% claim that I was never really comfortable with, I've never been really comfortable with the creationist explanations for this. I'm like, okay, so there is some evidence for a fusion. Why could there not have been a fusion? Why is it impossible? Now, if we look at the chromosome banding patterns of the great apes and humans, you'll see that they line up very nicely across most of the chromosomes. Now, some things have been moved around, but there's a lot of what's called synteny, or the same gene order, or in this case, stripe order, amongst the chromosomes. If you look at just human chromosome 2, which is on the top line here, you can see that the chimpanzee, which is the next two down, there's two chromosomes that line up with human chromosome 2, and they overlap at the ends. And there's two gorilla chromosomes and two orangutan chromosomes. The striping patterns are beautiful. Now, we're told that what created human chromosome 2 was an end-to-end fusion of two ape chromosomes. The problem is it's not an end-to-end fusion. The two ape chromosomes are too long. You have to lose a couple million letters there. Something got deleted. So this was a very complex thing. And so if things got deleted, it's a mystery of why the ends of the chromosomes are supposedly in human chromosome 2. but there is some evidence for that being there. In fact, if you take antibodies with a fluorescent tag and you paint them on chromosomes, you can light up all sorts of different things. And if you use a tag for what are called the telomeres, the ends of the chromosomes, it lights up in the middle of human chromosome 2, as if there is telomeric DNA inside chromosome 2. Now, it has been pointed out by several creationists, there's other places in the human genome where we have end-to-end telomeric sequences like this, and these aren't fusion events. How do you explain that, Mr. Evolutionist? But that's another story. I'm just saying that there is at least some evidence for this. In fact, another measure is called bias GC clusters. These are very, very common at the ends of chromosomes. So if you string the entire human genome out and you look at where these things are found, they're all found at the ends of chromosomes, the left and the right end of a chromosome. The only exception to that is in the middle, where that red arrow is, of human chromosome 2. Oh, so was there a fusion or not? Well, looking at Jeff Tompkins' work, which I really do appreciate. I appreciate this a lot. But he had this image in 2020 paper. This might have been Tompkins and Bergman, or I don't remember which paper it was from. But the image has two ape. In fact, but it's not. He's comparing the chimp chromosome to the human. There must have been a prior ape-like chromosome that fused to make the human in the evolutionary model. But we have chromosome 2. That pinch in the middle, that is a centromere, which is highly repetitive DNA. On the ends are the telomeres, which is a different repeat, but it's also highly repetitive DNA. And in chromosome 2 of human, if two ape chromosomes fuse, we should find a fusion site, which is the ends, and we should find a cryptic centromere. Now, honestly, this is a massive problem in that things like this tend to produce sterility or death, but if your chromosomes don't line up, you can't have children. If the eggs and the sperm and the DNA isn't alignable, then disaster happens in the embryo. So if this fusion happened, this thing must have settled down really quickly in order to allow this to propagate throughout the human species. But a new paper came out. This is on bioRxiv, partitioning the genomic journey to becoming Homo sapiens. Very interesting paper. It's just still sitting, it hasn't been published fully yet. But the paper is claiming that there are two things that are distinctively human in the genome. And that is the chromosome 2 fusion. I'm not saying it happened, but there is evidence for it. And then the pseudo-autosomal region 2. PAR, the pseudo-autosomal region, that's the tips of the X and the Y chromosome. You have to be able to line up your chromosomes. The Y and the X, they have to line up in the cell, and they line up at the tips. The claim is that the right-hand one is PAR 2, broke off the X chromosome and got welded onto the human Y chromosome, and that made the human Y chromosome distinct from all the apes. So there's those two things that separate apes and humans. The fusion of chromosome, the created chromosome 2, and the unique PAR2 on the human Y chromosome. But this paper is also hinting that all archaic humans and modern humans are just subpopulations of Homo sapiens. Ooh, very interesting. Because Neanderthals and Denisovans and all archaic humans ever sequenced have the chromosome 2 fusion and have the PAR2 on the Y chromosome. So these are distinctively human things that separate them from the apes. And hey, if you want a graduate level thesis, you creationist scientists out there who's looking for an idea, that might be something to look at. But I want to bring up another paper. It's Deacon 2018. They're looking at chromosomal differences amongst the marsupials. There is a theoretical marsupial ancestral genome, and you can color code different parts of the chromosomes. And then if you look at the different groups of marsupials, you see that the colors are found on different chromosomes. They have different chromosome numbers, and the colors are found in different places. So clearly there's been a lot of changes. If you look at the bottom set here, this is the opossum. There's a set of colors and chromosomes and lengths. The next one up, though, is a Tasmanian devil. The next one up is a wombat. The next one up are the macropods. That's koalas, not kangaroos, sorry, it's sort of okay. Kangaroos, wallabies, and tree kangaroos. But it's not all the macropods. Actually, this is 3 species of wallaby. Oh, they have different chromosome counts, and the pieces of chromosomes are found on different places in their genome. I think it's fair to say that every creationist would consider them one created kind. All the wallabies. In fact, I think all creationists that I know would lump wallabies and kangaroos and tree kangaroos into the same created kind, the macropods. That means that within this created kind has been a lot of chromosomal changes over time. So that raises the question, how come it could not have happened in humans? In fact, Karen Bettinger at the Proceedings of the International Conference of Creationism in 2013 had a paper called Chromosome Number Changes Within Terrestrial Mammalian Families. And she wrote, chromosome number diversity is widespread across families. At first glance, roughly 3/4 of all terrestrial mammal families contain members with variability in their karyotype form. Most creationists would put the family level as the created kind. Therefore, there's karyotypic diversity, in other words, chromosomal rearrangements within them. Just putting that out there for you. Now, regarding the fusion site itself, Tompkins and Bergman, 2011, published this, and I remember seeing this saying, wow, that looks like a fusion site, but their conclusion was it wasn't, because the ends of the chromosome has a very characteristic repeat. It goes something like CCC-TAA. Well, the reverse of that would be TTA-GGG. So the reverse means that at the end of the chromosome, you get one reading. I think I just said that backwards, but I think you get my meaning. And at the beginning of a chromosome, you get the reading in an opposite direction. If they join together, you should get backwards ones and forwards ones, just like we see there. But it's a very short segment. You're talking like, millions of letters got reduced to a few hundred letters. And all the repeats, well, they're not perfect. They're obviously scrambled and changed, and man, would have to have changed very quickly. But that does look like an end-to-end, and yet it doesn't explain what happened to all the other stuff that must have been deleted. Now, Tompkins, as he's defending himself, he did point out that there is a gene that spans the fusion site, a tightly regulated, highly expressed, very important gene that one of the exons, or the coding region for the gene, is on one side of the fusion site, and the other exons are on the other side of the fusion site. So this is part of the integration question for the evolutionists. Yeah, if we had a fusion, you have to integrate this into the systems so you don't die. And here we have a tightly integrated gene that spans the fusion site. Now, of course, the retort, this is associated with the telomeres, Tompkins, and blah, 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 blah. And there's all this arguing going back and forth, and I'm not going to get all into that right now. But again, there's evidence and counter-evidence and evidence and counter-evidence, and it seems like we go in circles. So I want to answer this more fully. How do we do that? Well, what if we had better data? What if we weren't stuck looking at, humanized chimpanzee chromosomes or questionable sequence reads coming out of a machine? What if we had a beautiful, high-quality, full-length genome with no gaps and no missing areas? Yes, we're in the realm of the telomere-to-telomere genomic sequences, finally. Last summer, more than 20 years after the completion of the human genome was finally completed. That is, they were able to sequence through the repetitive regions that were bedeviling them for all this time. We've invented something called long-read sequencing technology, and now we can read millions, or maybe 100,000 easy, but maybe a million letters in a row and go right through all the problematic areas. Be able to sequence the centromeres, the telomeres, all the gappy areas, all the repetitive areas. And about 6 months after the human was finished, Evan Eichler's lab, which is the people who generated the data that Tompkins was using earlier, but the Eichler lab came out with the chimpanzee, the gorilla, the orangutan, 2 orangutans, and the gibbon. And because they use a long resequencing technology, they could tell one chromosome from the other, to be able to split it into haplotypes. That is, what the father gave the chimpanzee and what the mother gave to the chimpanzee. This is amazing and beautiful data. And so it's also high quality in that they use a long read sequencing technology to cover the genome like 100 times over, which means you average out all the errors. And then short read sequencing technology another few hundred times. This is like the most coverage I've ever seen in any genomic study. The original chimpanzee genome was like 5-fold coverage, which meant on average they missed a bunch of spots and errors propagated into the data. No longer true. But the first thing I need to do is I needed to find the fusion site on chromosome 2, the so-called fusion site. But the data aren't annotated. Like the old genomes, you can say, okay, I want to find this. You can look it up and say, oh, it's here. But this data, I didn't know where anything was yet. And so I started searching. I searched chromosome 2 for the sequence TTA-GGG, and I found it all throughout the entire chromosome. Then I searched for CCC-TAA, the opposite, and I found it all throughout the chromosome. So I took the stuff from Tompkins' paper and I searched for TTA-GGG, TTA-GGG, and I found it in the black dots in the image in only certain places. And then I searched for a longer piece and it lit up a certain spot. And I said, that is where the fusion site is. And then I started doing lots of comparisons. I'm going to be using a program called BLAST, the Basic Local Alignmented Search Tool. It's extremely useful. You can do all sorts of things that the designer didn't intend for it. In fact, I've talked to him several times. He's complimented me a little bit on some of my penetrating analyses, but I'm using this BLAST program. And I started by saying, okay, let's just see how long the matches are between human and chimpanzee. What's the longest perfect match that I can find? So I went to random places in the human genome, I pulled up 1,000 letters, and I blasted it against the chimpanzee genome to see what it would find. Now the minimal match has to be at least 22 letters, but on the left here, this is human versus chimp, I found most of the matches were 50 letters long. Even though I was using 1,000 letters to search, in that 1,000 letters, it was only able to find an exact match of 50 letters on average, or in the majority. Now, one reason for that is, think about it. If we're 98% identical, that's 2 changes per 100. That means 1 change per 50. That means the average length of exact match would be about 50. Oh, okay, so that's kind of what they would expect. Fine. But then I did chimpanzee to chimpanzee. That is the 2 copies of the chimpanzee. And I saw the average length of match was longer, and I got some very long matches because of what's called identity by descent. I think this is the chimpanzee Clint, who's a famous research chimpanzee at Xerxes Lab here in Atlanta. And he's a lab-bred chimpanzee, so it's been several generations, and I don't think, I think he's got some homozygosity in his genome. I don't know how much, but his parents were not distant related, shall we say. But that's kind of what we get, pieces. Okay, step 1. Step 2, I now want to know, I know where the chimpanzee chromosome 2 is and where the fusion site is, and I need to line it up with chimpanzee chromosomes, but which ones? So I did 500 searches of 1,000 letter pieces of the human genome. I just picked them at random from different places in the human genome, and I threw them against the chimp genome to see where they stuck. And just look at the top left corner here. See that green square? Most of the human chromosome 1 sequences lined up with chimpanzee chromosome 1. But I also got some down in going the other way, chimpanzee to human, going down. Some of the chimpanzee sequences lined up with chromosome 18, 19, and 21 in human. In other words, the best match was in a totally different chromosome. But you'll notice on that second row down, human chromosome 2 going across, oh, look at that. The darkest green is chromosome 13 or chromosome 12 in the chimpanzee. Now, a long time ago, somebody decided and published a paper, hey, everyone, we shouldn't call these 12 and 13. We should call them 2A and 2B because we know they match with human chromosome 2. And when we do it in all our databases, 2A should be backwards. So it's the only chromosome in all the genomic programs ever that's loaded in backwards on purpose, and they renumbered them. I really like 12 and 13, and the original data from the Eichler lab, they had 12 and 13. The paper that they published, they changed the names to 2A and 2B. Too bad. But looking at this, there's a clear similarity between those two chromosomes and human chromosome 2. So I wrote a computer program. And this computer program ran for a long time. And what it's doing, it's going through a chromosome. Which way is it going? Human, chimpanzee, chimpanzee to human. I don't remember at the moment. I'm getting confused. But I'm going 1,000 letters at a time through the entire chromosome, maybe 10,000 letters at a time through the entire chromosome in one species and throwing it at the other species. But if you look at the numbers, I'm getting 99.1%, 98, 99.4, 98.7. That's a high similarity. But the length of the match, 2,000 letters, 3,000 letters, 1,600 letters. Out of 10,000, only about 2,000 are aligning. That means that, yeah, I get 99% identity across 2,000 letters, but the other 80% must have a lower similarity than that. Ooh, that's interesting. Now, if I take this program and I use two non-homologous chromosomes, like chimpanzee chromosome 11 and human chromosome 2, the matches are random. They're all over the place. It's just random, and this is the best matches. It doesn't mean a good match. It's any match. The best match of a piece of one chromosome thrown at the other, it can be found anywhere. But if I compare that, also the human chimp 11 to human 2, I see pretty low percent identities, 95, 96%. And this is cut off. There's a bunch of stuff on the left. There's a long tail to this data. There's a lot of matches less than 95%, but I get about 95, 96% match and 1 to 2,000 letters usually. Okay, now let's take that and let's change this. Instead of chimp 11, let's use chimp 12 and 13. In fact, it was called 2A and 2B, either way. And let's put that onto human chromosome 2. And so instead of seeing a match like this, what do you think you're going to see? That. Oh, yes. Chimpanzee chromosome 12 and 13, or 2A and 2B, align beautifully on human chromosome 2. Oh, and notice that the start of chimpanzee chromosome, this one here, the chromosome 12 is backwards. Chromosome 13 is forwards. Notice there's a very poor alignment here. This is the centromere of human chromosome 2. The centromere actually lines up better with chimpanzee chromosome 13 and chromosome 12. There's a little blob here of the deactivated centromere, the so-called deactivated centromere on human chromosome 2. But notice here, the ends of the chromosomes don't really line up on the ends very well at all. There's telomeric sequence all throughout human chromosome 2. But there's a big blob of it supposedly around the deactivated centromere. Oh, this is called synteny, or gene order, and it's very profoundly strong. So was there a chromosomal 2 fusion or not? Now, like the couple slides ago, if you look at the percent identity of chimp chromosome 12 versus human chromosome 2, we get a lot of 98, 99%. We get a lot of it. But we also get some less than that. So I'd ask you, if you want to take the average of that, how would you do it? Well, that's not easy, because this is also not normally distributed. It's not like a clear average with everything's varying around the mean. No, there's a long left-hand tail here. And in fact, I cut it off at 95% because I just didn't want to show all of it. It's really hard to take an average of this. This is going to take some pretty sophisticated statistics. Just saying, this is not a trivial thing. Now, looking at this, when I'm comparing these sequences, I can find places that match, but then there's places that don't match. In fact, most of this is gaps, because remember, this 10,000 letters, only maybe 2,000 of it match, so I put a dot there, but that means 80% of that dot is actually missing data. So I wrote another program. In this program, I start at one of those matching areas, and I look to the left and I look to the right, 1,000 base pairs at a time. Boom, 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 until I get to a place that has a poor match. And then I go to the left, boom, 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 until I get to a place that has a poor match. Oh, and there's a place right there that has a poor match. Terrible match. this data, as you notice, there's some underlines and hyphens in there. Those are spaces. Those are gaps that the program adds to keep the alignment going. Because when you compare two genomes, there's invariably going to be a single letter, a 2-letter, a 5, 10-letter gap where something's missing. And if you don't account for that, everything downstream is going to be messed up. So we have to add gaps to keep them going. Now, as I'm doing this, I'm extending things and I'm connecting these areas and to make longer pieces. I found something on I think this is the X chromosome. This is an area, this is 600,000 letters in a row of beautiful synteny, and the average match is about 98%. So yes, creationists, the human and chimpanzee genome contains very large sections of beautiful sequence that aligns. This is true. Now in the middle of that sequence, there are some spots that have terrible match. But if you keep going past the matched area, the match drops off to oblivion. Down here to 60%. But you know what that is? That's fake. It's not 60% similar. No. Remember, to keep these things aligned, you have to add gaps. So if I also show you the number of gaps that I had to add to keep them aligned, a lot of gaps to get that 600,000 letter sequence. But in the areas that don't align, lots and lots of gaps. Because if there's an A, oh, that doesn't match with the T in that gene. Oh, but if I add 6 letters gap here, I can get an A to line up. And if I add another 3 gaps here, and I can get this to line up. So you can do lots of gaps to keep something aligned, but 60% is artificial. If I look at the matched lengths of initially, lots of 2,000, 3,000, 4,000 things out of 10,000. But after I applied my gap filling technique, I got sections that were 160,000, 150,000, one over 600,000. Long pieces of DNA with very high similarity. My cutoff was 80%. So if anything dropped to 80%, that's when I stopped. I don't know what's there. I don't know what that sequence is. I don't know what's after that sequence. That was my metric, 80% cutoff, and I tried to fill gaps in to get to those 80%, and this is what I'm getting. Long pieces separated by areas of no similarity. How do you put a number on that? That's really hard. This is I think that's 23 million letters in this chart. Percent identity is probably 98%ish. The average length is 197,000 letters long. That's pretty significant. But again, they're separated by things that don't match. So this brings us back to my initial idea. We need more than one measurement. In the creationist community, we need to have a discussion. Was there a fusion? And when did it happen? Well, if there was a fusion, it had to happen before the Tower of Babel. In fact, I'm guessing it had to be on the Ark. In fact, I'm guessing all of Noah's family members had to have this fusion, or maybe they would have been reproductively incompatible. Or there'd be something that you'd expect a non-fused thing to be out there somewhere if someone on the Ark, which is 1/6 of the world's population, Shem, Ham, Japheth, and their 3 wives, only 6 of them. Yeah. When would it have happened? And did it happen? Also, I want to know what the actual percent similarity is. Just as I don't know, just as a nerd, I want to know that number. What is the similarity of the areas that we can align? It's high. What's the similarity when you add in the areas we can't align? It drops pretty far. How many mutations separate us? I want to know that. That's just an interesting intellectual exercise. I don't believe the evolutionary timeline, but I would like to see the numbers anyway. But then, I think we need to be discussing an integration model. That is, after a new gene arises, or a duplication, or translocation, or inversion, or anything like that, how does that gene become integrated into the system? That is extremely hard to answer. Literally, I'm not going to go there. I don't have time in my life to do that kind of work. That is a graduate level program, maybe even a laboratory level program with multiple people. That integration model. How do you get a new mutation to arise and become integrated into the system of complexity? Oh man, my head wants to explode when I consider that. But I think this way because of creationism. Evolutionists, and I'm making them mad just by saying this, but they have downplayed the complexity of life from day one. Always, life has been simple, and the more complex life becomes, the harder it is to explain. They had a good run of it for a long time, but they have run into the hard wall of complexity. Life is not simple. The selfish gene concept, I've seen Richard Dawkins being attacked lately. Old ideas, the one gene, one enzyme hypothesis, all these things that were their guiding paradigms for so long are destroyed in the modern world of technology. And yet, humans and chimpanzees are similar. How similar is a question that we're going to have to answer. Now, if you're a glutton for punishment, you can go to my website, biblicalgenetics.com, look up one of my articles. You can find all these also on YouTube. I've loaded some videos on Rumble, but I'm like 2 years, 3 years behind, so I wouldn't even bother going there. The other alternative social media platforms, YouTube, and some of these are on Facebook. They're there if you're interested. If you like genetics and want a really interesting presentation, check out my new DVD, Ancient DNA, Illuminating the Tapestry of Biblical Human History. Now, I know a lot of people don't have DVD players today, but you can stream this off creation.com, or if you get the disc, there's a QR code on the disc. If you put your phone over it, it'll add this product to your creation.com account, and then you can watch it on creation.com. Just saying, we're trying to cover all our bases here. But what I'm doing is I'm talking about the real Dark Ages, not the time before the Enlightenment. No, that was a brilliant time of human advancement. But the real Dark Ages is the centuries after the flood, as people are spreading out across the world, before civilizations are developing, before writing starts. Almost all of early human history is opaque to us because people weren't writing down what was happening, or if they did, we don't have the records. And so most of the migrations and invasions and wars and things are only in the DNA of the bones of the ancient people buried in the ground. Oh, that's a fun story. You can pick that up, get it off creation.com. Or I have a brand new book, Biblical Biology 101. It's the first book I've ever written with only my name on the cover. I'm so excited about it. What I'm not excited about is the typos that I know are going to appear. Yeah, some people have written in, "Hey, Rob." I was like, oh, man. But even after all, we had a dozen eyeballs read this. There's still a few typos in there and little mistakes and accidents. But second printing, all that will be cleared up. Right now, though, I'm very, very happy about Biblical Biology. It is beautiful. It's probably the nicest book that Creation Book Publishers, which is our publishing wing, ever produced. You can get it. Go to creation.com, scroll down, you'll see a link. Order now, Biblical Biology 101. And it is beautiful. I mean, the artwork, the idea development. This was a multi-year project, both in writing and in design. And it is, I'm just very, very, very proud of this. And I think you'll like it. In fact, if you order it and you put a little note in the sale thing, the secretary might bring me one to sign for you. I've signed a lot of them already. Just getting that out there for you. So there you have it. Reassessing human-chimpanzee genetic similarity. The answer is not easy to come by. There's a lot of information out there. It's really hard stuff. But honestly, it also doesn't matter because I can take it either way. That doesn't mean my God is not the creator. Okay, that's it. I hope you enjoyed that.
Oh, thanks, Rob. That was wonderful. I know you're just scratching the surface, but I appreciate the hard work you've done behind the scenes. And I know that sometimes we discover something new and fascinating and we kind of finalize it and move on. Obviously, in this area, it's a work in progress.
Yeah.
You're doing the work. So a lot of people don't like to say anything until they're done. So this is nice that you're along the way, sharing what you know for now, and you'll continue to refine it. I've always appreciated your demeanor and the way you come across. I think that's very important for us as creationists to be kind and to be Christ-like. We are ambassadors, and so we don't need to be angry about anything or do name calling. So anyway, appreciate how you conveyed all that. Very intense. And again, but I appreciate you trying to summarize as much as you can. I'll take a few questions. We won't stretch it out too long, but a number of questions have been come in. So I think think Zach's going to put some questions up to begin with. I can start out with one that I see that came standing. The truth is chromosome 9 has a telomeric like hexamer sequence, and its reverse complement arranged head to head
Yep.
in a similar fashion, but purported fusion site. But isn't this due to a fusion? Thoughts?
That's exactly what I was referring to earlier, and thank you, because I was literally read your paper just recently and watched part of your video on this again, just to refresh my mind on it. Yeah, there's telomere-like head-to-head stuff in the genome that shouldn't be there, and there it is. So it's not abundantly clear that this chromosome 2 is a fusion event. It's not necessarily so. I mean, or God fused it and left it there like that. I mean, I don't know, or it happened in human history. I'm just trying to ask the creationist community to have a more open mind, because we know these sorts of things happen in mammals. And there is an opportunity for it to happen in humans, and that's in a flood-like thing. Even for the evolutionists, they're saying this happened at a time where the human population collapsed down to maybe 1,000, 1,200 people. That's a near extinction event. That's a very small population. Well, that's also our model, my model anyway. If it happened, big if, it would have been a flood-like time frame for it to happen.
Sure. Thanks. Next question is going to have to do with junk DNA. So, Chuck, it says, do evolutionists still believe in junk DNA? Today we saw it mentioned in a 2024 article that did not discount the idea that it was worthy of the name junk.
It depends on who you talk to for the answer. Some people say absolutely, most of the human genome is junk. Some people, no. And you have to dig down into the history of why the idea was developed in the first place. It partly in response to something called Haldane's dilemma. Haldane was a very famous evolutionist back in the 40s and 50s, and he reasoned that natural selection could only act on maybe a few hundred letters in all of human versus chimpanzee history, and therefore you can't get that much evolution. And so 10, 15 years later, Ono and Kamura and Japanese scientists, they said, oh, well, what if most of the genome is non-functional? What if only, the 3% of the genome that codes for protein, that's what natural selection on. The rest is evolutionarily neutral, and we have neutral theory come in now. And then in the 70s and 80s, oh, look at that, it's all junk. Well, I just had an article on creation.com last week, the dark proteome. A brand new paper came out. It shocked me, because about a year ago, I read a paper where in fact, I did a video on this on on biblical genetics. Someone reanalyzed the number of genes in the human genome, and it went down. The initial estimate was about 22,000 and went down to about 19,000, which is about the same number of RNA genes. That's pieces of RNA that have introns and exons and alternate splicing and things, but they're never made into protein. So there's about the same number of protein coding genes as RNA genes. And then this new paper about the dark proteome, they found more genes than currently exist in the so-called junk DNA. Small genes, things that were lost in the noise, but produce proteins that are very functional and very important. Little teeny proteins, and there's tens of thousands of them. So yes, yet another class of junk DNA that has bitten the dust. But that doesn't mean there's no non-functional DNA. It doesn't mean there's not broken genes. It doesn't mean that there's not mutated genes. And it doesn't mean that there's not genes that don't actually have a function, or DNA that doesn't have a function. Part of that is because DNA has a three-dimensional shape. It's actually a four-dimensional shape, because the chromosomes change shape over time, the fourth dimension. And even though, say it this way, when the human genome was published, one of the first papers I read was they looked at genes that are in biochemical series. So genes that we know are used together to make something, make something else, and to get the end product. And they looked at their locations in the genome, and they found out that they're randomly distributed across the genome. And they said, oh, it's just junk. Millions of years of evolutionary leftovers and baggage, and genes are just randomly scrambled. And then 5 or 10 years later, scientists started treating the cell with formaldehyde. And formaldehyde causes cross-linking in the DNA. And then if you chop up the DNA, you get all these DNA X's. And if you sequence the arms of the X, you can see which genes are next to each other in 3D space. And they found out that the genes that are used together are next to each other in 3D, even though they're on different chromosomes. They're actually in spatially oriented, so that that so-called junk DNA that doesn't have a function, part of its function is spatial orientation. So the sequence might not matter, and it might not even the exact length of sequence might not matter, but the fact that that sequence is there is important. Cut out one of it, one piece of junk DNA, nothing. Cut another piece, nothing. But if you cut out enough of it, you'll kill the organism because it's not junk. But creation.com, just type in junk DNA. You'll get several articles that I've written, several other people on that subject. Great question.
Thank you. I would throw one of my own really quick because Haldane's dilemma was mentioned. Assuming for now that a high percentage of the DNA is not junk, how valid today is Haldane's dilemma?
It's on steroids.
Okay.
It's worse than it can be, which is why a lot of the arguments are trying to poo-poo our creationist stuff. It's not designed. It's just random. Trying to hold on to that junk DNA argument as long as they can. But that has also brought up other forms of evolution, evolution 2.0 and things like that, because they realize that the 1900s version of evolution actually doesn't work, and so they're looking for alternate ways of getting evolution to occur.
Great. Another question to put up. Question is, isn't DNA similarity irrelevant now because gene expression is what really counts? And is there a way to measure common gene expression?
Absolutely correct. And that's what I said. It's a gene expression between the humans and chimpanzees that's vastly different, not necessarily the sequence. Yes, there are ways of measuring gene expression, but that is not my field. I know that there are maps of which genes interact with which other genes and how they interact, but I don't do any of that work. And so the information's out there, but I don't know very much about it. So I'll leave that to you, Paul, to explore on your own. Maybe you can write a paper for one of our journals and tell us more. Cool question, though.
Right. Another question, does shared gene order, synteny, have a common design answer? Synteny is important for gene expression.
Yes, synteny is important for gene expression, especially when you start folding up those chromosomes into 3-dimensional shapes and 4-dimensional shapes. You can't just change it willy-nilly. And if there's a common designer and God's working on a common motif, he said, okay, I'm going to create life. I'm going to create eukaryotes. Now I'm going to create chordates and vertebrates and mammals, and now primates. And we are built, humans are built on a primate-like frame. We just are. We're very similar to them. It's like he said, okay, here's my generic thing. I'm going to make chimpanzees, gorillas, orangutans, and humans, but I'm going to put something extra in humans. And what's extra in is that je ne sais quoi, that spiritual aspect of us that apes simply do not have, not even remotely close. And he put enough genetic diversities between us so that we could not hybridize. Thank the Lord for that. But synteny is important on a programmatic level. But there are a lot of differences in synteny between humans and chimpanzees. There are tens of thousands of genomic rearrangements that you have to account for between the two species. That's a lot. Some of that's in, non-important areas, but there's other places where there's very important genes that are there or not there. I know this is old, but the first gene count I saw maybe 10 or 15 years ago, I think the number is 689, something like that. That's the number of genes that humans have that chimpanzees don't. Entire gene families that humans have that chimpanzees don't have. I don't know if that's held up with the long resequencing technology and stuff like that, but that was the original statement from last decade. There's a lot of genes that we have that they don't have, therefore synteny is not perfect. And if you're going to start changing that order of things, you're going to start changing those interaction networks, and that's complicated enough that my brain couldn't do it. It would take someone smarter than me with a lot more time and a very determined effort to figure out those pathways.
Thanks. I think we got a couple questions left, but I'm going to throw in one more myself because I can do that because no one's stopped me.
Okay.
And there's probably not an answer to this, but do we have any idea? All cellular life they do similar things. The cell has to reproduce itself, has to repair itself, has to dispose waste, it has to convert energy. They have all that.
Have you been reading the first chapter of Biblical Biology 101?
Yep, I will soon. Definitely. So there's a lot of commonality, so there's got to be high commonality in similarity in the DNA that does that. Do we have any idea in general what percentage that might be versus things that aren't basic cellular functions?
Oh, wow. That's a cool question. There are a set of genes that are shared across the entire animal world. I mean, it was actually a surprise when we found out that sea squirts have a lot of genes that we thought were only going to be in vertebrates. So there are general housekeeping genes that are very, very common amongst all living things. And how do we say this? The more similar things are, I'm not saying closely related, but the more similar things are, the more general genes they share. But there are other genes that are shared so widely across such a giant taxonomic spectrum, it's actually shocking. And some of those genes are extremely similar, like they hardly mutated over, hundreds of millions of evolutionary years. Mr. Evolutionist, those things can mutate. How come they didn't? Because they're not at old. But anyway, I don't know the number. I might have learned it once in a class. But I'm not sure what the number is.
Okay.
It's probably the majority of genes are shared widely. Because most cellular processes are common. Energy, reproduction, cell division, those things are common to all life forms. So we probably share most of those genes.
Sure. Thanks.
Except that things like reproduction, DNA copying, protein manufacturing between eukaryotes and prokaryotes is radically different. There's no evolutionary link between those two things, but if you're at least in the kingdom level, you're sharing a lot of genes. Let's say it that way.
Mm-hmm. Sure. Well, two questions left. One is, can we explain why chromosome differences between chimps and bonobos from an arc ancestor in 4,500 years? Looks like it's mostly gene loss. Similarity is 65.3, and the human chimpanzee is 26.2.
When the problem is that the Y chromosome is incredibly variable. When we sequence Y chromosomes, most of the sequencing work has been done on sections that don't vary very much. You add them together, it's a little over 10 million letters. But that leaves 55 million letters of the Y chromosome unsequenced in most projects. But when we look at humans, what is it, 20%? I was on either Grok or ChatGPT just last week asking these questions. I don't remember what the answer was, but there's very large percent differences from one human to another on the Y chromosome. Because the Y chromosome, there's duplications, inversions, and deletions, and all these massive changes that happen a lot. And humans and chimpanzees, chimpanzees are missing the entire heterochromatic arm of the Y chromosome. And then when you look at the parts we do share, the similarity is really low. So can we explain chimpanzees and bonobos, the two very similar species that I'm going to put in the same created kind as basically the chimp created kind? Yeah, because you're not talking about sequence specificity. You're talking about multiplication and deletion of large pieces of DNA, which can happen very quickly. If a Y chromosome suddenly loses 1 million letters, that's a pretty significant percent difference, but it's only one mutation. And it can happen, and it does happen in that particular chromosome. I wish you and I, Donnie, could actually talk about that right now, but that's my best short answer. And by the way, everybody, I can't see the comments, so Jay surprises me with all this. I I got
Sure.
the private chat window open, which is good because I wasn't distracted by seeing comments stream by. So
Yeah, there were a lot of very positive comments and a lot of going back and forth with questioning things from
okay,
people who are coming from a different angle, which is great. So one question left, other than I got one of my own, just something
okay.
for you to comment in general, and it could be series of programs, but the concept of orphan genes.
Ah, yeah, orphan genes are amazing. Um, there were unexpected, shall we say, that there are taxonomically restricted genes. There are genes that only humans have. There are genes that only platypuses have. Well, that's not too unexpected. There are genes that um, very small species groups have in very large pools of things that there hasn't been enough time for those genes to evolve in that little group or that one species. In fact, at every taxonomic level, we see brand new genes that don't have homology or common ancestry with others. And that's a puzzle for the evolutionists. Now, some of them, they say, oh, it's incomplete lineage sorting. That this thing was back here, but all the other groups might have lost it, and only this little group here kept it over time. Okay, that's possibly an answer for some of it, but orphan genes are an enigmatic thing for the evolutionary community, and yeah, of course they have answers, and people bluster, people talk loud, but I have not heard a satisfactory answer from them on that topic. But that's what an orphan gene is. Is literally it's orphaned. It shouldn't be there, and they're everywhere.
Sure. Okay, great. The last question, I don't can't tell if it's just wanting clarification or surprised that you said something.
Hmm?
Said, did Dr. Carter just admit substitution rate is different from mutation rate?
I'm gonna, yeah, I'm gonna guess who asked that. Oh no, not the person I thought asked that. Um, okay, this brings up a very interesting thing in standard population genetics. If you have a well mixed population, and it actually even doesn't matter the size, the mutation rate over time will approximate the substitution rate. That's you combine two population genetics formulas and you get that. So whatever the mutation rate is, that will be the long-term substitution rate. And that's one of the reasons why the evolutionist has a way of explaining in 6.5 million years humans and chimpanzee differences, at least a lot of them. The problem with that is, I said, well, mixed population. We know the human population today is stratified. We know that in the past it's been stratified. Um, Random mating is pretty much a really bad assumption. That's not human behavior and doesn't fit geography either. But there's something else very interesting. The human race has not evolved in 10,000 evolutionary years. Zero evolution. Why? Because we've been going through an exponential growth spurt in the evolutionary model since the dawn of agriculture about 10,000 years ago, and in exponentially growing populations, you get no fixation. If you That's the substitution rate. In other words, a mutation rises in some farmer in India, and that has to spread throughout the entire human race. So every single person now has two copies of that mutation. It has been fixed. The original thing was lost. That's required for a species to evolve. But humans have not evolved since we started expanding. And also, the substitution rate will, or the fixation rate, will slow to a crawl in very large populations. If a mutation occurs, it would take a fantastically long time to spread throughout the entire population. Now, given maybe 8 billion people, and everyone has about 100 new mutations, we'd expect the fixation rate to be about 100 per generation, but we have an exponentially growing population, so the fixation rate is zero and has been for a long time. So, in other words, whatever your population model is, you can simplify it by assuming random mating and assuming a lot of things. Or you can realize that throughout human history and chimpanzee history, lots of changes in population size and stratification and separation have occurred, so the number isn't nearly as easy to come by as a simple formula might indicate. How's that?
That makes sense. A
Okay.
lot of sense. Well,
Great
thanks
question,
again,
though.
Rob. Yeah, no, great questions came in. I appreciate anyone who's taken time to listen, no matter what side of the fence they're on. We appreciate all listeners. It's how we challenge each other. And again, I appreciate, Rob, all the great research you've done for many, many years. And I'm looking
Thanks.
forward to here in future programs that we will do with you to see updates on things, getting more data. So as a reminder, make sure you guys check out our website, which is logosresearchassociates.org. Consider supporting one of the many research projects that we're working on. Also, make sure you subscribe to Logos Research Associates email so that you can receive notifications about our future and upcoming programs. And you'll also get put on the list for Creation Evolution Headlines, which is just great information scouring the literature of articles that are out there related to the Creation Evolution controversy by David Coppage. And then the recording of this will be available to watch it again later or to send links to other people. So our next presentation is going to be Joel Tay. He's going to be talking about a biblical view of dinosaurs, which sometimes can sound silly, like, why are Christians even talking about dinosaurs? But most Christians not only don't know what they believe about dinosaurs, they don't know what they're supposed to believe. So it's just kind of a weird thing, but it's a great topic when you realize that the Bible has a lot to say about dinosaurs. And the more we look at the science, the more it lines up with what Scripture said all along. Who knew that maybe God knew what he's talking about? Even though the Bible is God's first shot at writing a book, I think he did a pretty good job. So that's our next presentation. And again, Rob, thanks for taking your time being here with us. Again, we look forward to seeing you on future programs.
Well, thank you, everybody. I really appreciate your time.
And I will just close in a word of prayer, and then we'll see you in the next broadcast. Dear Heavenly Father, just thank you again for this time, and thank you for Rob's presentation and all we can learn for it. Sometimes when you go deep, we can lose some of the audience, but there's a lot of people who we can't go deep enough for. They want the details. So we're thankful for the brilliance you've given to a lot of scientists out there, and we thank you for revealing your truth to us in your word too. And we thank you for sending your son again to die on a cross to pay for our sins. That's more important than any scientific details we could possibly talk about. So we just thank you again for this time and pray all these things in Jesus' name. Amen. And we will see everyone next broadcast.